The project in SR&D this year involves mutations. The genes that Al likes to study have recently been shown in publications to have some mutations that are then also associated with some diseases. So what we're going to do is we're going to try and recreate those mutations and express them in a cell line so that we can see what those proteins are doing in a functioning cell. The way we're going to do this is using quikchange mutagenesis, which was developed by the Strategene company, and this is their graphic explaining how it works. They used green and yellow to represent sort of the template or parent DNA that does not have a mutation, and then the blue and the purple are the primers that have been created. The X's are the mutations. That would be a single base that is not correct. It does not base pair to the template, but they do base pair to each other, these two primers. So it's basically done like PCR, where you denature the DNA using the high temperature, and those two strands are going to come apart. Then you cool it down so that the primers can anneal, and then you allow extension to occur using a polymerase to add DNTPs and go around the circle the rest of the way. So that in the end, you have a almost complete blue circle that will pair with an almost complete purple circle, and then the two parent strands could come back together. So the parent strands were made in E. coli, in bacteria. And when bacteria make copies of their DNA, they methylate it. So this has methyl groups stuck all around it. There's an enzyme called DPN1 that will digest methylated DNA. So if you add DPN1 to your entire PCR reaction, it should digest this into teeny little pieces, leaving your PCR product uncut. So what we need in order to do this is the template plasmid that has the cDNA for your gene in it, not mutated. And Al already has all of these. Then what you're going to do, Al's written a protocol where you're going to go onto a website and design the primers with the mutation that you need. He'll order them, and then we're ready to do PCR. So the primers that you're going to design will partner, I don't know what that was, will base pair with the plasmid like so. Let's say your mutation needs to be right here, so they will pair there. So I thought it might help if you could see sort of what that would look like. So here we have our plasmid that's going to act as our PCR template. The primers have the mutation in them, so the purple down here has the mutation, the green does not. Well, when you put it in a thermal cycler, first thing that's going to happen is those two strands are going to separate. So we're going to get this strand separated from this strand. And then when you cool it down to the annealing temperature, the primers can anneal. So maybe this primer anneals here, and this primer anneals here. And then you heat it back up a little bit to the elongation temperature, and then the elongation is going to happen around this way where we're adding new DNTPs, but it can't close the circle. It can't connect in that direction. So we just sort of have they call it a nicked circle. And then this one's going to do the same thing. And I'm going to have a nicked circle here. Well, then I'm going to heat it back up. I'm going to denature it again. And now these two strands come apart. And I've got more primers. And so this primer maybe goes here. And this primer maybe goes here. But now I have these two nicked circles that also can have primers anneal to them. So like this primer could go here. And this primer could go here. So then I'm going to cool it back down and let it go to the elongation temperature. And I'm going to get a nicked circle here. And this one's going to make a nicked circle. And this one. And this one. And you're going to do 30 cycles of this. I'm not going to draw 30 cycles of this. But then when you heat it back up and it denatures, then all these come apart. But then in the end, when you cool it back down, they can anneal to each other. And so here I've got a nicked circle and a nicked circle, and then here, let's see if I can make both of these move at the same time. Put these together, and then I'll go ahead and put these back together. So this would be your end product after just a few cycles. This is methylated DNA. So I don't want that. It's the template. It doesn't have mutation in it. So you digest it with DPN1, and it will cut it apart. And so now you've got these nicked circles that have your mutation sequence in them. Now there's two ways people have handled the fact that these are nicked circles and not complete circles. Option number one is that you can use a ligase enzyme. So you can add ligase enzyme and it'll come in and connect all the circles like this, like this, like this, so that you don't have nicked circles anymore. Option two is you can just go ahead and transform. For some reason, E. coli is kind of okay with it having some nicks in it, and as soon as it gets inside, it's like, hey, there's nicks, and it seals those nicks by itself. But either way, you now have your gene with the mutation that you designed. And that is how we're going to introduce your mutation into E. coli.